Hello friends, I am Sunil sir and I am back with a new video. Friends, in this video I will be discussing about very important aspect, that is how to score full marks in spotting. Normally you get 10 marks for your spotting, and if you know your spotting very properly, you can easily score full marks. So this video is totally dedicated for the biology practicals, that is spotting. For spotting, you will be given 1 marks each from spot A to I, that is 10. Half marks will be for identification when you identify that spot, the name. Another half marks is to write something about that spot. Now something means you have to write at least 2 points. We have divided the spotting into two parts, botany and the zoology. From A to F will be all your botany spots, and from G to J will be all your zoology spot. So let's start with the spotting and watch the video till the end so that you understand each and everything for spotting, and out of 30 marks of your practical, you can easily score 10 marks. The first spot that we have is name the plant and comment on the floral adaptation for the type of pollination. Now, when we talk about floral adaptation, if they provide a diagram, something like this, maize related diagram, then we need to understand maize is not at all an attractive flower. It is not bright, it is not colorful, it does not produce nectar or scent. So it is showing which pollination. The maize plant is going to show wind pollination. So it shows floral adaptation for wind pollination, and it is called as anemophily. The flowers are unisexual and the plant is monoecious, having male and female flower on the same plant. Male inflorescence is at the top, called as tassel, whereas the female inflorescence is axillary, which is called as the ear. Female, the flowers basically they are small, they are not attractive, they are not bright, not colorful, nectarless. They have perianth which is reduced to two locules. The anther is versatile. Pollen grains are light in weight and large in number. This is all you have to write when you get maize for spotting. But at the spot A, instead of maize, they might keep sometime colored flower. So name the plant and comment on the floral adaptation for type of pollination. When there is a colored flower, or you can see insect and the flower diagram, then you need to understand it is insect pollination. And the example for this is salvia. Salvia shows lever mechanism. Flowers are adapted to insect pollination, that is entomophily. Salvia flower, bisexual, they have to be attractive, bright, colorful, so that they can attract the insect for pollination. When we talk about the nectar, the flower produces nectar. The nectar and the nectar glands, they are present. They are situated in such a way that when insect tries to reach the nectar gland, its wings and the body part will definitely touch the anther and the stigma. Salvia shows biplepped corolla. The larger lips encloses style and stigma. There are two stamens located at the mouth of corolla tube. The pollination mechanism that is seen in case of salvia is liver mechanism. So when you see a bright colorful flower or the insect and the flower, you have to remember it is entomophily, insect pollination. Flower has to be bright, colorful, attractive. They must produce nectar and scent. This is what you need to write if you get colored flower on spot A. Now on spot B, there will be a question, identify and comment on the stage of meiosis. Now here, they might provide you a diagram like this. Looking at this diagram, you have to identify which stage is this. This is anaphase 1. When I say anaphase 1, how I understood it is anaphase 1, not anaphase 2. The reason behind this is, look at the chromosome. The entire chromosomes are moving towards the opposite pole. In anaphase 2, the chromosome will break from the centromere and then they will move towards opposite pole. So when you look at the chromosome, if it is complete, then it is anaphase 1. It is an important stage of meiosis 1, as reduction, division takes place in this stage. That is, chromosome number is reduced to half of the total number. Spindle fiber starts condensing, becomes shorter, and it pulls the chromosome towards opposite poles. It results in separation and dragging of recombined homologous chromosome towards opposite poles. When you look in the slide, it will appear like this. This is anaphase 1. Now, they might keep some other stage of meiosis also. Like, look at this stage. It's showing two nuclei. It means it is telophase 1. In telophase 1, you will say the chromosome reaches towards the opposite poles. Nuclear membrane and the nucleolus, they reappear. Chromosome will recoil to form chromatin material. Spindle fibers will disappear and they form two daughter nuclei. So when you look at this diagram, you can easily understand this is telophase 1. If they want to show telophase 2, they will keep 4 nuclei, not 2. Next, spotting C, identify and comment on the technique. It is experiment based and you have to identify. So they might show you diagram like this. What exactly it is showing? So you can see a forceps here, stamen is removed. So it is very clearly indicating it is emasculation. What is emasculation? Removal of anther before anthesis. And this emasculation is a step of hybridization experiment. Emasculation is done to prevent self-pollination. Emasculation is the removal of young anthers of stamen in the plant selected female parent well before anthesis, that is formation of pollen grain. Bisexual flower of plant which is selected female parent is emasculated, means male part is removed to avoid self-pollination. Buds are open and the stamens are removed mechanically. It can be done by the help of forceps or by given hot water treatment or even by the help of scissor. On spot C, if you don't get the forceps and the flower, you might get something like this, a plastic bag tied to a flower or a plant. So it is indicating again a step of hybridization experiment, which is bagging. So emasculated flowers are then bagged in polythene bags or sterile paper bag to prevent pollination by undesired pollen. So bagging is always done so as to avoid the unwanted pollen grain that is present in the air to fall on the stigma and do pollination. For emasculated flower, always bagging is done. The desired pollen grains are then dusted on the matured stigma of emasculated flower by opening the bag temporarily. So bagging, at the same time you can see a small tag also attached. So the upper experiment is for bagging, but there is one more experiment that can come in C category is identify and comment on the technique. So here you can see a tag, a label has been attached. Tagging is done as a part of hybridization experiment. Tagging is done just to remember all the details of the experiment because it is not possible to remember each and every characteristic of that particular plant or the experiment. So tag is tied to the bag. The tag carries brief information about the names selected parent varieties, day, date and time of emasculation, anthesis and actual crossings are recorded on that tag. So you can see a tag here. Tagging is the part of hybridization experiment. The spot D, it can have name the plant and comment on its xeric or aquatic adaptation. So on spot D, either you will get those plants which are adapted to xeric condition, that is desert condition, or the plants which are adapted to aquatic condition. So this plant, when you see such kind of plant, it is called as Calotropis procera, also known as Rui. It is aquatic? No, it is desert adaptation, a xeric adaptation. It is non-succulent xerophyte. The leaves are thick and leathery. The plant contains milky latex. If you break the leaf of this Calotropis procera, you will see white sticky fluid coming out. That is Calotropis. Next, what you can get on D, if not Calotropis, you can see something like this. When you look at these thorns, it appears like babool. So here it is Acacia arabica. The common name is babool. Big thorns, small leaves are the identifications for Acacia arabica. It is a non-succulent xerophyte. Non-succulent means it will not store any water. Leaves are bipinnately compound. Leaflets are very small in size to reduce transpiration. The stipules are modified into spines. The reason behind spine is two. One, to avoid transpiration. Second is to protect themselves from grazing animals. Or on spot D, you can get name the plant and comment on its xeric or aquatic adaptation. Here you can see very common plant, that is cactus. It is xeric adaptation. In exam, you have to say the given spot is Opuntia dilini. That is not funny. It shows xeric adaptation. It is a succulent xerophyte. It means it stores water. Leaves are modified into spines to reduce the rate of transpiration. Stems, they are modified into phylloclad to perform photosynthesis and store water. In this plant, you will not find leaves. Why? Just to avoid loss of water by transpiration and at the same time protect from browsing animals. On spot D, you might also get some aquatic adaptation. Look at this plant. Looking at this plant, most of the time students say it's a type of animal. No students, it is plant, aquatic plant. It is hydrilla. It is a submerged hydrophyte. When I say submerged, it means it is well below in the water. The stem is very soft, slender, without any mechanical tissue. Leaves are arranged like whorls. Cuticle and stomata are absent because they are in water. The entire plant is covered by mucilage just to avoid osmosis. Or on spot D, you can get name the plant and comment on its xeric aquatic adaptation. So if you see something like this, it is aquatic adaptation, and this is well-known plant Eichhornia water hyacinth, also known as terror of Bengal. It is a free floating hydrophyte. It floats. Stem is modified into offset. It is spongy due to parenchyma. It has air present in the parenchyma's tissue. Petioles are swollen due to air chamber. Roots are adventitious and they have root pockets. Root hair is absent. So all this you need to write when you say Eichhornia on spot D. The next spot again on D can be one more. This is a type of adaptation in plant. It is typha, also known as cat tail. The tail of the cat appears like this. It is amphibious anchored hydrophyte. Again, it is aquatic adaptation. Stem is rhizome with adventitious root. The leaves are long, linear, soft and spongy. Leaves have mechanical tissue, hence they can stand erect. Cuticle and stomata is also present on the leaves. Now coming to spotty, they might give you a test tube like this, and inside there will be a paper, and based on this you have to identify that this experiment is of separation of plant pigment by paper chromatography. Since it is for spotting, you don't have to write much for this spot. You will write aim to study the separation of plant pigment by paper chromatography. Then you will write the observation. The chromatography paper strip shows four different colors, distinct band from the loaded spot: yellow green, bluish green, yellow and orange. Each color represents one pigment. So let's see what you will write in the conclusion for this spot. You will say the pigment travels at different rates along the chromatographic paper on the basis of their molecular weight and solubility in the solvent. Chloroplast of spinach leaves contains four different pigment. If there is chlorophyll A, yellowish green, chlorophyll B is yellowish green, chlorophyll A bluish green, xanthophyll yellow and carotene orange. This is how the pigments get separated. When you look at this diagram, you will understand the first pigment to get separated is chlorophyll B because of its higher molecular weight, which is yellowish green in color. Then chlorophyll A, which is bluish green in color. After chlorophyll A, the pigment which gets separated is xanthophyll, yellow in color, and at the last, the lightest pigment which gets separated is the carotene, which is orange in color. Remember the order, it is B, A, X, C. The next spot that you can get is spotty is rate of transpiration by four leaf experiment. Now in this spot, four leaves will be tied and it will be shown and you have to identify. So whenever you see four leaf, just write, it is very clear, it is rate of transpiration by four leaf experiment. What do you need to write, students? You need to write the aim of the experiment. Aim is to study the comparative rate of transpiration in upper and lower surface of leaf by using four leaf experiment. What will be the observation? Observation on leaf A. Here there will be Vaseline applied on both the surface, so it looks fresh and dry. On leaf B, The Vaseline is applied on the lower surface, so it is slightly dry. On leaf C, Vaseline is applied on the upper surface. It is more drier than B. And on D, Vaseline is not applied at all. It D means it dries completely. So this is what you have to understand. Now, conclusion that you can derive. On leaf A, I will say Vaseline is applied on both the surfaces, therefore no transpiration occurs and leaf is fresh. On leaf B, Vaseline is applied on the lower surface because we all know that maximum stomata, they are present on the lower surface of the leaf. So very less transpiration occurs as the number of stomata is less on the upper surface. Therefore leaf is slightly dry. On leaf C, Vaseline is applied on the upper surface. So more transpiration will occur because maximum stomata is on the lower surface of the leaf. Thus the leaf is more dry than B. And finally D, where you have not applied Vaseline on any side, the transpiration occurs from both the surface and the leaf has dried up completely. You can see in the diagram also A, B, C and D. Look at the D diagram. It is totally dried up. The next spot is spot F. Identify and describe. Here students, you will be given specimen. You have to identify. Now this, when you look at this diagram, it's very easy to understand. This is anatropous ovule. On spot F, if you get something like this, you have to write it. This given specimen is for BS of anatropous ovule. It is the most common type of ovule found in angiosperms. Anatropous ovule has a bent axis with micropyle directed downwards. It is covered by two integuments. It consists of embryo sac, female gametophyte. Embryo sac consists of egg apparatus, two polar nuclei and three antipodal cell. Or even you can say it is 7-celled and 8-nucleated structure. On spot G, you might get disease causing organisms and comment on the symptoms of the disease caused. So here you have to talk about symptoms. Spot G, identify the disease causing organism and comment on the symptoms. If I show you this, here you can see mosquito. The moment you see mosquito, just say this is plasmodium. It causes malaria. And symptoms of malaria includes fever, shivering caused by release of hemozoin, arthralgia, joint pain, vomiting, anemia caused by hemolysis. Cyclic occurrence of sudden coldness followed by rigor and then fever. Sweating lasting for 4 to 6 hours, occurring every 2 days. These are all the symptoms of malaria. You don't have to write the life cycle, you have to write the identification and the symptoms. Again, one more disease that you have to identify. Instead of malaria, if they give a diagram something like this, then you have to identify students that this is Entamoeba histolytica. Entamoeba histolytica is responsible for causing amoebiasis in man. Symptoms include amoebic dysentery, abscess in liver, lungs and brain, passing out stool with blood and mucus, abdominal pain, nausea, bowel irregularity. So these are all the symptoms of Entamoeba histolytica which causes amoebiasis. If not this on spot G students, then on spot G you can get one more specimen of this. Looking at the diagram, you can understand this is Ascaris lumbricoides. And this Ascaris lumbricoides is responsible for causing ascariasis. What are the symptoms? The common symptom is vague abdominal pain. In severe cases, the patient may experience restlessness, weight loss, anorexia, distended abdomen, intermittent loose stool, and even occasional vomiting. Let's talk about spot G. If not Ascaris, then you might get something like this. This indicates ringworm. Even though it is ringworm, it is not caused by any worm, student. It is caused by fungus. So it's a fungal disease which infects hair and feed on the keratin protein. It causes dermatomycosis. Symptoms of ringworm are red ring patches, lesions on the skin, intestine itching, there will be and scaly skin. So huge itching or intense itching can be seen. Coming to the spot H, it is all related to human brain. Name the part of the brain and mention its function. So for that, human brain has to be studied. If they show you diagram like this, they might ask you the bigger portion is called as what? So it is cerebrum, largest part of the brain. It has four lobes. Which are the four lobes? Frontal lobe, parietal lobe, occipital and temporal lobe. Students, you have to name the part and you have to mention the functions. Don't mention they have sulci, gyri, sulcus, not required. They are only asking the functions of cerebrum. So the function of frontal lobe is controls voluntary activities. It is related with memory, intelligence, problem solving, judgment, reasoning, etc. Parietal lobe is responsible for controlling sensation of touch, pressure, pain and temperature. Temporal lobe controls centers of smell, hearing and speech. Occipital lobe controls sense of vision. Now name the other part of the brain. It can come on spot edge like this. So this red color area that you can see, it is nothing but cerebellum. So it is the second largest part of brain. Maintains equilibrium, posture, balancing. It is the center for neuromuscular activities like walking, running, etc. If not, then name the part of the brain mentioned functions. First one was cerebrum. If not cerebrum, then you might get cerebellum. If not cerebellum, then you might get this. Name this part which yellow arrow is pointing. It is nothing but medulla oblongata. It controls all the involuntary vital functions such as heartbeat, respiration, peristalsis. It controls non-vital activities such as coughing, sneezing, swallowing, vomiting, yawning, and sudden injury to medulla oblongata can lead to death. Let's move on to spot eye. Here you have to identify and describe the permanent slide. Now this slide will be seen under microscope. So this, when you observe under microscope, you will say that this is TS of testis, transverse section of testis. Each testis contains 200 to 300 seminiferous tubules. Seminiferous tubule is lined by cuboidal germinal epithelium. It shows different stages of spermatogenesis, which are spermatogonium, primary and secondary spermatocytes, spermatid and sperm. Few large pyramidal cells also is seen in between the germinal epithelium. Those are called as Sertoli cell, or even they are called as nurse cell. They provide nourishment to the sperm till maturity. You don't have to write all the 4 points, at least 2 points is sufficient enough. But identification is very important for TS of testis. When you observe under microscope, it will appear violet color with lobules. So this is TS of testis diagram under microscope. Let's talk about, if not TS of testis, then what else can be seen in spot eye. Spot eye, you will see something like this. This is nothing but the ovary. TS of ovary, or transverse section of ovary. Internally, the ovary shows compact structure with outer cortex and inner medulla. The medulla shows connective tissue called stroma. The cortex is lined by germinal epithelium. Different stages of developing ovarian follicles are seen in the cortex. As the follicle grows, it forms secondary and mature Graafian follicle. One ovum from the mature follicle is released from an ovary in every menstrual cycle, after which mass of yellow cell is formed. It means after the release of the egg or ovulation, a yellow colored body is formed called as corpus luteum. If corpus luteum is there and if the egg gets fertilized, corpus luteum will remain. But if the egg is not fertilized, corpus luteum degenerates to form a white colored body called corpus albicans. But if the egg gets fertilized, corpus luteum will remain and we can say the lady is pregnant. Under microscope, students, this is how the TS of ovary will occur. If not TS of ovary, under spot eye, first take a look. TS of ovary, Graafian follicle under microscope. So this slide, if you see, you have to remember it is TS of ovary or Graafian follicle. If not TS of ovary, then you might get this. It is blastula, which is also known as VS of blastula. It means vertical section of blastula. So under spot eye, students, you might get TS of testis, you might get TS of ovary or VS of blastula. It shows outermost small flattened cell layer called trophoblast. It encloses a cavity called blastocele and inner mass embryonal knob. The inner cell mass is attached to the trophoblast on one side. So the cells of trophoblast which are touching the inner cell mass, it is called as cells of Rauber. Under microscope, how it will appear, it will be like this. You can see the cavity present in the center, that is called as blastocele cavity. Let's move on to the next spot, that is spot J. Spot J says identify the animal and comment on its xeric and aquatic adaptation. So students, you might get something like this. Now you can easily identify this animal is camel. It is a xerocele animal adapted to desert condition, excretes concentrated urine because it has to conserve water. It lives in desert. It has long legs. It accumulates fat in the hump so that heat flows away from the body and inward flow of heat is prevented. It has long eyelashes, nostril hairs that protects the eyes and the nose from sand dunes or strong winds. Next that you can get under J, if not camel, you can get something like this. This little cute animal is kangaroo rat. This kangaroo rat can survive its life without water. It means it is showing xeric adaptation. It is a xerocele rodent, nocturnal. It comes out mostly in night to avoid heat of the day and to have humid air inside. Kangaroo rat neither sweat nor pant to keep itself cool. Next specimen that you can get under J is this. It's very commonly known aquatic adaptation in animal. It is a fish, that is rohu. It is a freshwater fish. Body is laterally compressed and streamlined in order to minimize resistance of water and thus reduces friction with water. Presence of gills for respiration which helps them in exchange of gases in water. Body is totally covered by scales to prevent osmotic entry of water in the body. It has a caudal fin that helps as a steering. It helps in changing the direction of the fish while swimming. The next spot that you can get on J, if not camel, kangaroo rat or rohu, you might get this. This is well-known mammal, aquatic mammal perhaps, that is dolphin. It is an aquatic mammal, body is streamlined, smooth and furless to reduce friction. The posterior and you can see it has a caudal fin that helps in swimming. The position of the nostril near the top of the head, it facilitates easy breathing when animal reaches the surface of water for breathing. Dolphin, remember students, is a mammal, an aquatic mammal. It has flippers for steering, slowing and going up and down or out of water. Hope students you would have understood the spotting that will come in your exam and this might have helped you a lot. If this video has helped you, don't forget to give a like, don't forget to subscribe to the channel if you are new. This is your Sunil sir who will be bringing biology in easy way for you. Let's meet in the next module with some new topic. Thank you very much. Study hard, stay blessed.